After a polypeptide chain is formed, it's going to be folded into its secondary and tertiary structure into a very specific 3D conformation or shape. And at this point, we can start calling it a protein. But this protein may not be ready to carry out its function just yet. There might be some additional protein modifications that need to be made to this protein before it can be functional. And those are called protein modifications. There are two different types of protein modifications. The first type is co-translational modification. And that means that these are modifications or changes that happen to the protein, or actually to the polypeptide, while it's being translated. So let's say we have the ribosome right here, and we have a polypeptide that's being formed. So these changes are going to happen while the polypeptide is being formed. And an example of a co-translational modification is acetylation. And what happens during acetylation is the first amino acid in the polypeptide, which is usually methionine, is going to be removed. And in its place, we put an acetyl group. So let's just draw an acetyl group. And acetylation happens to 80 to 90% of eukaryotic proteins, but the significance of this modification is not known very well. We're actually trying to figure out what the purpose of this modification is. The other type of protein modification that happens is post-translational modification. And actually, most protein modifications fall into this category, and the examples we're going to discuss in this video are all post-translational modifications, and those modifications happen after translation. Many post-translational modifications happen in the endoplasmic reticulum and the Golgi apparatus, but not all of them. So let's go through some examples. So the first post-translational modification I want to talk about is glycosylation. And glycosylation, well, you can look at the word, the prefix glyco tells us that it has something to do with a carbohydrate. And so glycosylation is the adding of a carbohydrate to a protein. And most of the proteins in this video are all in green. And glycosylation usually happens to proteins that end up being embedded in the cell membrane. So you can see here we have a cell membrane and this protein embedded in it, and then there are these carbohydrate groups attached. So here's a carbohydrate group attached, and here's another one. And glycosylation helps to identify different types of cells. And one very common example of where we use glycosylation is in the ABO blood groups. So let's take 4 different red blood cells, and let's just say that each one of these red blood cells belongs to a different person. And so red blood cells have these proteins embedded in their surface. And these proteins are going to have many times different carbohydrate groups attached to them. So let's say that this person right here has this particular carbohydrate group attached to it. That makes him blood type A. Let's say that this person has a different type of carbohydrate attached to the protein. Let's say it looks something like that. That makes them blood type B. Let's say that this person has actually both of those carbohydrates attached to his red blood cells. That would make him blood type AB. And let's say that this last person does not have any carbohydrates of this category attached to the proteins on his red blood cells, and so that makes him blood type O. So here's a very common example of how glycosylation is used in the identification of different types of cells. Let's go on to a different type of post-translational modification that's pretty similar, and that is lipidation. And lipidation is when we add a lipid to a protein, also a protein that's going to be attached to the cell membrane. And this lipid we're looking at is actually an example of a GPI anchor. And GPI anchors are lipids that help to attach or tether proteins to the cell membrane. And just to give you an idea of maybe why this would be necessary, so to quickly review the structure of the cell membrane, we have these hydrophilic heads. That means that they are polar. And then we have inside these hydrophobic tails. And that means that they are nonpolar. And so the protein has both polar and nonpolar parts on it, and maybe it just doesn't attach well to the hydrophilic portion of the cell membrane. And so this GPI anchor, a lipid, kind of plunges into the lipid or hydrophobic part of the cell membrane, and we know that substances that are similar, like substances that are both hydrophobic, attach very well to each other. So this lipid, which is hydrophobic, attaches very well to the inner part of the cell membrane that is also hydrophobic. And so that's how it helps to attach the protein to the cell membrane. And both glycosylation and lipidation usually do occur in the endoplasmic reticulum or in the Golgi apparatus. Let's move on to some protein modifications that have more to do with the activity or the function of the enzyme and less with the structure. So one very, very common protein modification I want to discuss is phosphorylation. And phosphorylation is basically the adding of a phosphate group to a protein or to an enzyme. And then phosphorylation comes along with dephosphorylation, is when you remove a phosphate group from the enzyme or the protein. And I'm just going to make a little bit of room over here. And so what you're looking at is this schematic diagram of the sodium-potassium pump that's found in basically every animal cell. So the sodium-potassium pump. And another name for the sodium-potassium pump, which is the enzyme that you're looking at, is the NAK-ATPase. And so this enzyme or protein, the sodium-potassium pump, is responsible for maintaining the proper osmolarity of sodium ions and potassium ions in and out of the cell. And so let's see how phosphorylation regulates this protein. So, and again, the proteins that you're looking at, they really all represent one protein. It's just we're going through the motions of the changes that happen to this one protein. But it's not like we have it's not like we're looking at 6 different proteins in the membrane. So here's our first step. And when you look at this enzyme, you can see that there are 3 receptor sites for a particular ion. This is the sodium ion, represented by these dark blue circles. And then there are 2 receptor sites that look kind of more squarish, and those are receptor sites for the potassium ions, and those are represented by these light blue squares. So those are the potassium ions. And so back to the first step in what's happening here. The sodium ions will attach to the receptor sites on the enzyme. And just to clarify, this is the intracellular space. So this is basically the cytoplasm. And so these sodium ions are coming from the cytoplasm, and then out here is the extracellular space, the outside of the cell. Anyway, back to our first step. So the sodium ions attach to the receptor site, and when the receptor sites are full, it's going to cause something to happen. It's going to cause an ATP molecule, adenosine triphosphate, to break down into ADP, adenosine diphosphate plus phosphate. And then this phosphate will attach itself to the protein. And that is phosphorylation. And so when this protein gets phosphorylated, when the phosphate group is attached to it, it causes there to be some sort of change in the conformation of the protein. And that change in conformation causes the protein to turn itself around by 180 degrees and face the outside of the cell. And that's what you're looking at right over here. So our protein is still phosphorylated. I know I drew it in a different place, but just, keep in mind that there's still a phosphate group attached to that protein. And then the protein releases these sodium ions into the extracellular space. So the next step is number 4, and again, we are still phosphorylated. There's still a phosphate group attached to our protein. And so in the next step, the potassium ions on the outside of the cells will attach themselves to the protein on the receptor sites. And take note, there are 3 receptor sites for sodium, but only 2 for potassium. When the potassium receptor sites are full, it's going to cause a different change to happen. This phosphate group is going to be removed, and that is called dephosphorylation. So that is dephosphorylation. And so that phosphate group ends up in the inside of the cell and it gets recycled in some other way. And when this protein is dephosphorylated, when that protein is removed, it's going to cause a different change in the conformation of the protein. And that change in conformation causes the protein now to turn around again by 180 degrees, and it faces the inside of the cell. So that's our 5th step. You can see the protein is facing the inside of the cell. And in the last step, the potassium ions are released into the inside part of the cell, and then we're back to our first step. And so you can see that the phosphorylation and dephosphorylation basically regulates the activity of this protein. And the end result that we're trying to get to is that we want there to be on the outside of the cell a rather low concentration of sodium, a rather high concentration of sodium, and a rather low concentration of potassium. And on the inside of the cell, we want there to be a rather low concentration of sodium and a relatively high concentration of potassium. And that's accomplished by the fact that for every 3 sodiums that are pumped out, 2 potassiums are pumped in. And so here's an example, a very common example of how phosphorylation regulates a protein. And this doesn't happen just in the sodium-potassium pump. It happens in many, many enzymes and proteins in our bodies and in cells. Let's move on to 2 other protein modifications that also have to do with regulating an enzyme or something similar to that. So the next protein modification I want to talk about is methylation. And in particular, the methylation of certain proteins called histones. And those are these green circles that you're looking at. And histones are these proteins that around which DNA wraps itself. So they're found in the chromosomes and they help to package DNA in a very tight and organized manner. And sometimes histones are methylated. So let's put some methyl groups on our histones. And methylating and demethylating histones helps to turn certain genes on and off. And so here's another example of a protein modification helps to regulate activity, but in this case we're regulating the activity of genes as opposed to proteins. And another protein modification I want to bring up is proteolysis. And by looking at this word, proteo means protein and lysis means to break something down or to cut something. And proteolysis is sometimes to take a protein and activate it, we need to cut it. And in fact, the insulin has to be cut twice before it's activated. So let's cut this protein twice. And you may have actually heard of the term zymogen. And a zymogen is an inactive form of an enzyme. And sometimes the way to activate a zymogen is by cutting it. And that's proteolysis. There is one more protein modification I want to discuss. And that is sometimes we add a protein ubiquitin, to another protein. So that protein I just added is ubiquitin. And this process is called ubiquitination. And what ubiquitination does is it basically marks this green protein for degradation or for breakdown. So within a short while of being ubiquitinated, this protein is going to be destroyed and the different parts are going to be recycled. Let's just quickly review the post-translational modifications that we discussed. So we talked about glycosylation and lipidation. And those are two protein modifications that generally happen to proteins that end up being embedded in the cell membrane. So glycosylation was the adding of a carbohydrate group, which helps to identify certain cells, and lipidation is when you add a lipid to a protein that generally helps to anchor a protein to the cell membrane. Then we talked about phosphorylation, methylation, and proteolysis. And these all had to do with activating or deactivating an enzyme or genes. So phosphorylation was we brought the example of the sodium-potassium pump, which is basically regulated with phosphorylation and dephosphorylation. Then we spoke about methylation, which can basically helps to turn on or turn off certain genes. And proteolysis is the way in which many enzymes are activated. And the last post-translational modification we talked about was ubiquitination. And ubiquitination marks a protein for degradation, and then the various parts are recycled.